Well guys, we'll go with the third concept of animal physiology and anatomy, that is called as body fluids and circulation. So what are the body fluids and how it aid in circulation is the concept what we are going to discuss. Majorly the body fluids that helps in circulation are A, blood, and B is lymph. So these are the two body fluids that we have which aid in circulation. So let us consider what are the various patterns of circulation that takes place in various groups of organisms. When we go with the circulation, usually various group of organisms follow various modes of circulation. When it comes to the lower organisms, the unicellular organisms, this circulation is aided by body surface, by simple diffusion or by endocytosis. When it comes to the simple multicellular organisms or complex multicellular organisms, a particular circulatory system is involved. Certain organs are involved for circulation. Certain organs are involved for circulation. So which includes the circulatory system. So now the question is, what are the various kinds of circulations takes place in the living organisms? So let us go with what are the different circulations that takes place in the living organisms. There are circulation that takes place in the form of one, single circulation, takes place in case of organisms like fishes. Incomplete circulation takes place in case of amphibians and reptiles. Whereas complete circulation takes place in organisms like reptiles, particularly crocodiles, as well as birds and mammals. So what do you mean by single circulation, incomplete circulation and complete circulation? We will discuss in the later concepts when we talk about this circulation. So now the question is, what do you mean by circulatory system and how this circulatory system help in circulating various substances that are necessary for the body? So circulatory system includes A, heart, B, blood, and C, blood vessels. Circulatory system includes, or circulatory system is a composer of three different organs, one heart, blood and blood vessels. So let us go with what are the various internal and external structures of each of this circulatory system. So what are the three concepts? One, blood. B, heart. C, blood vessels. So we will discuss individually regarding this components of circulatory system. So when we go with the first one, blood. So I have to mention you the body fluids, blood and lymph comes under fluid connective tissue. It is also called as false connective tissue, fluid connective tissue or false connective tissue, because it doesn't have fibers like collagen or elastin or reticulin in the matrix. Absence of fibers like collagen, elastin, reticulin in the matrix of the connective tissue. That's the reason why it is called as false connective tissue. So when we go with the first one, that is called as a blood. Blood is a fluid connective tissue with matrix called as plasma and consists of solid part or also called as a fluid part. Matrix is usually called as fluid part, and the solid part includes blood corpuscles. Solid matrix or the ground substance or the fluid matrix is a fluid one, which is called as a plasma, and solid part includes the blood corpuscles. So when we go with the matrix of blood, that is called as a plasma, it is around 55% of the blood. Plasma consists around 55% of the whole blood. So out of this, 92% alone consists of water, whereas 6 to 7% it includes organic salts. Organic salts. So what are the various organic salts that are present in 6 to 7% of the plasma? It includes blood proteins, like albumins, globulins in the form of immunoglobulins, which help in creating immunity to the body, creates immunity to the body. So which are IgA, immunoglobulin A, so also called as alpha immunoglobulin, IgG, immunoglobulin G, that is gamma immunoglobulin, IgD, delta immunoglobulin, IgM, mu immunoglobulin, IgE, epsilon immunoglobulin. So these are the various immunoglobulins that are present. So when it comes to the other blood proteins, it includes fibrinogen, okay, are some of the blood protein. Along with that we have heparin, which is an anticoagulant. Okay, and this all blood proteins are formed in the liver. Make it clear, these are formed in the liver. So when it comes to the next component, that is called as the solid part, which includes the blood corpuscles. Blood corpuscles are of different types. They are red blood cells, are also called as erythrocytes. Red blood cells or erythrocytes. The second are WBCs, are also called as leukocytes. Third, also called as platelets, are also called as thrombocytes, are the various kinds of blood corpuscles. So here I have to mention that the albumin accounts for 4% of the 6 to 7% of the organic salts, whereas globulins accounts for 2 to 2.5%. So these two you may be asked in your NEET. Okay. When we go with RBCs, also called as erythrocytes, the first of the blood corpuscles, that is called as RBC. So RBCs are also called as erythrocytes or red blood corpuscles. Red blood corpuscles. So why these erythrocytes are called as red blood corpuscles? Because within the RBC it holds or it nurtures a red color pigment, respiratory pigment, which is red in color. A respiratory pigment, red colored one, which is usually called as hemoglobin. So due to presence of this respiratory pigment that is called as hemoglobin, which is actually made up of a ferrous ion in its 2+ state and a globin. So this presence of ferrous ion gives red color to the hemoglobin. So that's the reason why this particular cells, erythrocytes, are usually called as red blood cells. When we go with the erythrocytes, erythrocytes are biconcave in nature. They are biconcave in nature. Okay. So if you see from its lateral sides, it gives a biconcave shape. But if you see from the other side, it is spherical in shape. Okay, and denucleated or enucleated. That means absence of nucleus. Absence of cell organelles. As there is absence of cell organelles, anaerobic respiration takes place in these cells. So, as the absence of cell organelles, there is no mitochondria. So, as there is no mitochondria, anaerobic respiration takes place. The cells in the living human beings or living bodies, particularly in mammals, where there is a nucleus is absent as well as cell-bound cell organelles are absent. Due to absence of the cell-bound cell organelles as well as the mitochondria, anaerobic respiration takes place. The cells where anaerobic respiration takes place in the body are erythrocytes or RBCs. Okay. So usually RBCs, which consist of hemoglobin, carry the oxygen. So hemoglobin have a formula of Hb4. So hemoglobin can hold 4 molecules of oxygen to form oxyhemoglobin. Oxyhemoglobin. And it is said that 1 molecule or 1 gram of hemoglobin, 1 gram of hemoglobin transports 1.34 ml of oxygen. 1.34 ml of oxygen. In such a case, we have to know that 100 ml of blood consists 15 grams of hemoglobin. That is 100 ml of blood carry 1.34 ml into 15 grams, which is equivalent to 20 ml of blood, ml of oxygen. 20 ml of oxygen. Okay. So as I was mentioning, they are enucleated, particularly in mammals, and there is an exception for this enucleated stage. One is camel and second one is llama, are the 2 mammals which are having nucleated RBCs. Okay. We should know that these RBCs are usually formed in various parts, in various stages of life. In fetal state, stage, RBCs are formed in liver. So that's the reason why liver is also called as hemopoietic organ. Whereas in adults, in human beings, so RBCs are formed in bone marrow. Red bone marrow. So the process of formation of RBC in red bone marrow is usually called as erythropoiesis. Formation of red blood cells in RBC, red bone marrow is usually called as erythropoiesis. And it should be known that this red bone marrow produces all the blood corpuscles like red blood cells, WBCs as well as platelets. So usually the lifespan of an erythrocyte is around 120 to 127 days, which is approximated as 125 days. Approximated as 125 days. The worn out or dead RBCs are destroyed in liver and spleen. That's the reason why liver is usually called as graveyard of RBCs. Graveyard of RBCs. So what is the count, blood count? RBC count. What is the RBC count in blood? It is counted as 4.5 to 5.5 millions in cubic millimeter. It is around 4.5 to 5.5 millions in cubic millimeter is actually the count of RBC in the blood. So usually RBC's plasma membrane is called as Downan's membrane. The membrane of RBC is usually called as Downan's membrane, and this membrane has permeable to bicarbonates, chlorine ions, whereas impermeable to sodium and potassium ions. Okay, this permeability and impermeability creates Hamburger's phenomena, which we will be discussed in the breathing and exchange of gases. And this phenomena is usually called as Downan's phenomena. Okay, Downan's phenomena. So the next one is WBCs, also called as leukocytes, Here these WBCs are divided into two different categories based on presence or absence of granules. Based on presence or absence of granules in the cytoplasm, they are categorized into two types. One, granulocytes, B, agranulocytes. So this granulocytes and agranulocytes, based on the shape of its nucleus, they are further divided into three different types. In case of granulocytes, they are acidophils, which can be stained in acid solution, with the acidic solution, are also called as eosinophils, eosinophils, what we call it as. The second one is basophils, and third one is usually called as neutrophils. So as I have mentioned that the cytoplasm consists of granules, and the nucleus present in these granulocytes have different shapes. When it comes to the acidophils, they have bilobed nucleus. If you take it as a cell and if you see it, it has a bilobed nucleus. When it comes to the basophils, they are having S-shaped nucleus, or having many lobes. And when it comes to the neutrophils, they are multilobuled. They are multilobuled. So each of these granulocyte has certain functions, and they are usually act as microscopic policemen, scavengers, as well as they act as a macrophages in killing the foreign antigens and foreign microbes. When it comes to this percentage, around 60 to 65% alone accounts for neutrophils, whereas basophils accounts for 0.5 to 1%. 60 to 65% alone accounts for neutrophils, whereas basophils is around 0.5 to 1% in the leukocyte count. When it comes to the acidophils, it is around 2 to 3% in nature. So what is their actual functions? So these acidophils help in protecting the body during allergic conditions. Protecting during the allergic conditions. And this particular basophils secrete various substances like heparin, histamine, serotonin, okay, each of them, this heparin act as an anticoagulant in the blood, histamine act as a precursor for creating allergic reactions, serotonin usually controls the circadian cycles, as well as it also acts as a vasoconstrictor in the digestion of the food material, whereas neutrophils are phagocytic in nature. They are phagocytic in nature. When it comes to the next two components, monocytes, when you go with the agranulocytes, there are again of two types. The first one is called as monocytes and the second one is usually called as lymphocytes. Monocytes are the largest WBCs, make it clear, the largest WBCs, whereas lymphocytes are the smallest WBCs with a large nucleus, whereas largest WBC with kidney shaped nucleus. That means they have a kidney shaped nucleus, they are agranular, the cytoplasm doesn't have any kind of granule. When it comes to the monocytes, monocytes are the smallest WBCs with a large nucleus covering the entire area of the cytoplasm. So that's the reason they are usually, not they are usually, but the lymphocytes, they have a major function of providing immunity to the body. Whereas monocytes acts as microscopic policeman of the cell, or it is also called as scavengers of the blood circulatory system. So when it comes to the monocytes, monocytes account for 6 to 8%. Sorry, it is 6 to 8%, whereas lymphocytes account for another 20 to 23%. 20 to 23%, and they usually provide the immunity. We can see here the percentage of each of this WBC: acidophils, basophils, neutrophils, monocytes, lymphocytes. You see that each of them have different proportion in the blood, but alone the lymphocytes and neutrophils account for around 80 to 85%. Okay, whereas other WBCs account for the rest of the proportion. Okay, now let us see what are the various accounts we can give it for WBC. So WBCs, as I mentioned you, they are also produced in red bone marrow. Okay, and they have amoeboid movements. They have amoeboid movements. That's the reason why WBCs can escape through the lining of the blood vessels into extracellular fluids. So its count is around 6,000 to 8,000 per millimeter cube, and their major function is to protect the body from foreign toxic substances, antigens, or microbes. Okay. Sometimes we can see that this WBCs increase its count in certain stages. So WBCs usually increase its count during infections with bacteria, viruses, etc. Whereas this particular WBCs, if it exceeds one lakh due to uncontrolled proliferation, it leads to tumors called as leukemia. It's called as leukemia. That is called as blood cancer. It's also called as blood cancer. And this condition is usually called as leukocytosis. This condition is usually called as leukocytosis, and decrease of WBCs is usually called as leukocytopenia. So these are the three important things that has to be remembered. What is that? During infections with bacteria and viruses, the count of WBCs increases. A condition is usually called as leukocytosis. If this WBCs exceeds 1 lakh due to uncontrolled proliferation, it leads to tumor in the blood cells, WBCs, which is usually called as leukemia or blood cancer. Sometimes we can see that due to the infections, these WBCs get declined in the blood, a condition that is called as leukocytopenia. Make it clear, whenever you use a term phenia, it is a decrease of the cells or decrease of the tissues or anything. Cytosis means increasing, proliferating. Okay, and leukemia is a disease condition where there is uncontrolled growth of this particular cells. Well, it is biconvex in nature. As I mentioned you, if they are nucleated, okay, as there is no hemoglobin, that's the reason why they are called as white blood cells. Are some of the important features of WBCs. So the third type of blood corpuscles are usually called as platelets or thrombocytes. Thrombocytes are also called as blood platelets. So when you go with the thrombocytes or blood platelets, these cells are produced again in the red blood cells by special cells that are present in the red bone marrow that are called as megakaryocytes. So megakaryocytes present in the red bone marrow produce this third type of blood corpuscles, which are usually called as blood platelets or thrombocytes. So I did not mention here the lifespan of the leukocytes. The lifespan of the leukocytes ranges from 2 to 8 days, and the second cells usually live simply for only hours. Okay. Why these blood platelets are called as thrombocytes? Because they have a protein present within them which helps in healing the repairing the blood vessels, that is called as prothrombin. So that's the reason why these are also called as thrombocytes. So when we go with the thrombocytes majorly helps in healing, repairing of the blood vessels. Okay, how it does? What is the constructive phrase we can give for healing and repairing the blood vessels? It is usually called as clotting of blood. Clotting of blood vessel. So what happens in this clotting of blood vessel and how it heals the repairing of the blood vessel? It includes this particular thrombocytes includes various blood clotting factors which aid in clotting of blood. It starts from, it is given in the form of Roman numbers 1, 2, 3, 4, 5, 6 is rejected. 6 is rejected. 7, 8, 9, 10, 11, 12 and 13. There are 13 blood clotting factors, out of which 6th one is rejected. The first one is fibrinogen. Second one is prothrombin, which is in inactive form. Third one is thromboplastin. Fourth one is calcium, which is necessary for blood clotting. Without this calcium, blood clotting doesn't take place. Almost all the blood clotting factors are necessary for clotting of the blood, but it is more important. Fifth one is proaxerlin, or also called as axerlin. Sixth one is rejected, I said you. And seventh one is proconvertin. Eighth one is also called as anti-hemophilic globulin. Okay, which is absent in hemophilia, a condition that is called as absent in hemophilic patient, absent in hemophilic patients, hemophilic A patients. Ninth is usually called as Christmas factor. are also called as thromboplastin. 10th one is usually called as Clot factor. 11th one is called as plasma thromboplastin. 9th and 11th one is called as antiplasma thromboplastin antecedent. 12th one is called as Hagman factor. 13th one is usually called as fibrin stabilizing factor. Fibrin stabilizing factor. So as I mentioned you, for clotting of the blood, usually these factors are necessary. All these factors are necessary, like fibrinogen, which is usually present in the plasma, which is actually a soluble form which convert into insoluble form, and usually frame a network along with the platelets to close the particular opening. Prothrombin is usually an inactive form of enzyme, and this get converted into thrombin, and this thrombin usually in presence of calcium ions give rise to formation of a clot. And calcium ions, as I mentioned, it is necessary for all the enzymatic reaction which act as a coenzyme. Thromboplastin usually which is a part of which is a part of the tissue where this thromboplastin along with the plasma thromboplastin produces an enzyme that is called as thrombokinase, and this thrombokinase is necessary to convert the prothrombin into thrombin. And this thrombin enzyme act on fibrinogen, and this fibrinogen get converted into fibrin, which is initially a soluble form, get converted into insoluble form. Okay. So what happens during the clotting of blood? So let us see what actually happens during the clotting of blood. And I have to mention there is this, all clotting factors are formed in the liver. So if this blood clotting process doesn't take place, what is going to happen to the body? Suppose you have a cut internally or externally, blood usually oozes out. So the blood which is oozing out again gets stopped initially within 2 to 8 minutes. The time factor is always 2 to 8 minutes. So whenever cut takes place, blood usually oozes out, and this oozed blood at the place of the cut again gets stopped within 2 to 8 minutes. That's the reason it is called as blood clotting time. So blood clotting time ranges from 2 to 8 minutes. So what happens during the blood clotting? Let us see it. So whenever the blood with the blood platelets flowing in the blood vessels identifies an injury, the platelets get accumulated at that particular area, and blood platelets produce an inactive form of enzyme that is called as plasma thromboplastin, which is usually the which factor? Plasma thromboplastin, which is usually called as Christmas factor, which is a ninth factor. And tissues give rise to tissue thromboplastin. And this plasma thromboplastin and tissue thromboplastin mix together, they unite together to give rise to an enzyme that is called as thrombokinase. So this enzyme, in presence of calcium ions, act on prothrombin in its inactive form present in the platelets. In presence of thrombokinase and calcium ions give rise to thrombin. Okay? This thrombin act on fibrinogen in presence of calcium ions, and thrombin give rise to fibrin, which is in its soluble form. This in presence of again thrombin and calcium ions give rise to fibrin, which is in its insoluble form, forms a network over that particular injured part and heals that particular wound. So this is how the blood clotting takes place in the blood vessels. So thrombocytes are anucleated blood fragments lifespan is very less, 1 to 2 days are some of the things what has to be discussed. Okay, so once again, blood corpuscles are of 3 types: RBCs, which are also called as erythrocytes, WBCs are also called as leukocytes, and blood platelets are also called as thrombocytes. As I mentioned we have discussed a variety of things, but the major function of RBC is to transport around 97% of oxygen in and out of the body. At the same time, the plasma aids in transporting the nutrients, hormones, antigens, enzymes, etc. from one part of the body to other parts of the body. Around 20 to 25% of carbon dioxide is also transported through the red blood cells in the form of carbaminohemoglobin, and as I mentioned plasma as well as our red blood cells are the major components of the blood which usually transport the gases like carbon dioxide and oxygen. Here, when it comes to the WBCs or leukocytes, WBCs are protective in nature, and we have discussed that each of this cell, either it is acidophil, basophil or neutrophils, monocytes or lymphocytes, each of them have a specific work. So acidophils are majorly involved in controlling the allergic reactions. When it comes to the basophils are major organs along with the mast cells to produce various kinds of enzymes like heparin, which is an anticoagulant, serotonin, hormones particularly serotonin, histamine, which also helps in creating allergic reactions. When it comes to the A third group, that is neutrophils, which are the maximum in number with 60 to 65%. They are usually helps in phagocyting various foreign antigens, foreign microbes, as well as toxins. When it comes to the what you call monocytes, monocytes are the largest WBCs of the whole WBCs, and they have a kidney-shaped nucleus, and its major function is acting as a microscopic policeman, tracing out the various kinds of foreign antigens and killing them. When it comes to the last group of WBC, which are called as lymphocytes, they are usually they are the immunological cells which usually aid in giving immunity to the body. The platelets, which are also called as thrombocytes, are majorly involved in repairing and healing the ruptures that are created in the blood vessels during the flow of the blood. As I mentioned you, they have different clotting factors from 1 to 13, and each of these clotting factor has a specific role in the blood clotting. But it is mentioned that blood clotting time usually takes place between 2 to 8 minutes. So when the during the clotting of the blood, you can see that during the process of clotting, you can see that the blood platelet release plasma thromboplastin, and ruptured tissues release the tissue thromboplastin. These two thromboplastins mix together to give rise to an enzyme that is called as thrombokinase, and in this thrombokinase is necessary to convert the inactive form of prothrombin that is present in the platelets to active form of thrombin, which also act as an enzyme for the next step that is called as converting the fibrinogen in presence of calcium ions and thrombin give rise to fibrin, which is soluble part, and this in presence of thrombin and calcium ions further give rise to insoluble form of fibrin, and this fibrin holds the various kinds of cells in that particular ruptured part and heals or clots that particular area, not allowing the blood to ooze out. That's the reason after a cut between 2 to 8 minutes we can see that blood clotting time takes place. It is mentioned that if one of the clotting factor is absent, what is the clotting factor? That is clotting factor 8, that is anti-hemophilic globulin. If it is absent, the blood cannot be clotted, and that's the reason why hemophilic persons, whenever there is a cut, it starts on bleeding without any kind of healing, and sometimes it may lead to the death of the person. Okay, thank you. We'll go with the next concept that is called as blood groups. As we are talking about blood, we have discussed about the plasma and the corpuscles that are present in the blood. We will discuss about blood groups. So what are blood groups? We know that when we go to a hospital, when we are asked for diagnosing the type of blood group, people come with different reports like A positive, A negative, B positive, B negative, AB positive, AB negative, O positive, O negative. So where it has started, this blood groups, how it has started? The first person to find out the blood groups was Karl Landsteiner. Was the person who first time framed or formed these blood groups, and he discovered the three types of blood groups that is called as A, B and O. Later, another scientist named as Di Castello and Struly have discovered AB blood group. So how these blood groups can be identified? Let us check it. Usually blood plasma holds antibodies for a specific antigen. and this antigen is usually a globin, globin part. And on the surface of RBC we have antigens. Based on these antibodies and antigens, blood groups are divided. So when we go with blood group A, other than that, on the surface of the RBC along with the antigen, there is one more antigen for which antibodies are absent in the blood plasma, is usually called as Rh factor. Rh factor. So there are two types of antigens in the blood. One, antigens, there are different types we will discuss. Along with that, there is one more antigen that is present on the surface, which is usually called as Rh factor. Presence of this or absence of this decides the kind of blood we have or kind of blood group we have. If Rh factor is present on the surface of RBC, it is usually called as Rh positive presence. Presence of Rh factor gives rise to Rh positive, and absence of Rh factor gives rise to Rh negative blood. But when it comes to the antigens that are present, in blood group A, if it is blood group, in blood group A, it is seen that antigen is antigen A, whereas the antibody will be B. Antigen A will have antibody B in its plasma. So this is on the surface of RBC and this is in the plasma. In case of blood group B, the antigen is B, whereas antibody is A. That is in RBC and plasma. When it comes to the blood group AB, antigens will be A and B, whereas antibody, absence of antibodies. And whereas in case of O, there are no antigens, whereas antibodies A and B are present in the plasma. So these conditions are necessary to find out the kind of blood. As I mentioned you earlier, presence of Rh factor, absence of Rh factor further divides these blood groups into two types. In presence of that, it is called as A positive. In absence of, if Rh is present, plus Rh positive, minus Rh, A negative. In case of B, it is called as B positive, B negative. In case of AB, it is called as AB positive, AB negative. In case of O blood group, it is, if present, it is called as O positive, O negative. So based on this, we can divide into 8 type of blood groups. It is seen that based on this compatibility, so whenever Rh antigen A is present in the RBC, always the antibody will be B. If the blood group with antigen A is provided with antibody A, agglutination takes place. Agglutination takes place. That means clumping of the blood takes place, which leads to the death of a person. So based on this compatibility, a disease condition is usually seen in the pregnant women who give birth to the babies. So let us find it out. So due to this incompatibility or agglutination, we can see that if a Rh negative mother marries a Rh positive father, the baby that is formed will be Rh positive. But during this period of time, what happened? The blood vessels or the blood of this mother will form antibodies against this Rh factor. Antibodies are formed in the mother for Rh positive blood. In such a case, the first pregnancy or first delivery will be successful, whereas due to the formation of antibodies against this Rh positive blood, the death of fetus or baby takes place in the second conception. To avoid this, medical technology have developed where the mother is given anti-Rh antibodies. Anti-Rh antibodies are given after 72 hours of the delivery to kill the Rh positive antigens. Okay, let us once again have a eye on this. This condition is usually called as erythroblastosis fetalis. Erythroblastosis fetalis is the disease condition where the first delivery of the baby takes place successfully, but during this period or due to the injury of the blood vessels, the mother develops Rh positive antibodies, and during its second delivery the baby usually dies in the second conception. To avoid these conditions, we have a medicine that is given within 72 hours of the delivery that is called as anti-Rh antibodies. Okay? And this disease condition of death of the baby is usually called as erythroblastosis fetalis. Once again, I am making you, if a Rh negative mother marries an Rh positive father, the baby may be having an Rh positive or negative baby. But during this period of time, antibodies for Rh positive antigen is produced in Rh negative mother, blood of the Rh negative mother, and it is seen that the first delivery will be successful, but when it comes to the second conception, the death of the fetus or baby takes place, and the disease condition is usually called as erythroblastosis fetalis. But this condition can be avoided by taking anti-Rh antibodies to avoid such death of the fetus or the baby.